everybody. I'm going to take you through some of the projects that we have at the SENS Foundation Research Centre, which we tackle together with our many collaborators over here from all kinds of different universities and companies. So SENS Foundation Research Centre also existed under the Methuselah Foundation, SENS Foundation's precursor organization, right? So back in 2005, basically, when we started, how do I advance slides?
Green button on your right.
Ah, there we go. Cool. In 2005, Aubrey de Grey had assembled this theoretical plan. There's my point. Okay, thanks. Aubrey de Grey had assembled this theoretical plan called Strategies for Engineered Negligibles, in essence a plan for curing human aging, listing all the different types of damage that build up in our bodies. And I was a very little fresh graduate back in 2005, and together with a whole bunch of other folks, we volunteered for Aubrey de Grey, and we decided, all right, well, let's just see if we can implement some of Aubrey's theoretical suggestions. So there we are, the ragtag crew of volunteers who wants to cure human aging. How do we get started? And in the end, we figured the easiest part of Aubrey's plan was the intracellular aggregates that we heard about this morning from Anna Maria, the proteinaceous aggregates and neurodegeneration. There are others. So the Aubrey's plan for addressing this problem was that humans don't seem to have enzymes to break some of these down. Well, it's partially true. In the case of neurodegeneration, of course, we do have the proteases, but for Anna Maria's reasons, they aren't being used. For other targets, such as lipofuscin or, for example, oxidized cholesterol, we don't have the right enzymes. And so that's the part that this plan is dealing with, to discover enzymes which break these things down, deliver those enzymes into human tissues using methods like Jeffrey's, and see if we can get rid of our storage problem. So this is the crew of the SENS Foundation Research Centre. Most of these, unfortunately, are part-time volunteers. We have only a few paid full-time scientists. And that's our lab, a warehouse that we converted into a biotech laboratory in Arizona. The problem is catabolic insufficiency in aging. I just took you through this. These are the big 4 examples of intracellular aggregates, other aggregates, catabolic insufficiency, neurodegeneration, we heard in detail from Anna Maria, diabetes, where you get these advanced glycation end products that modify extracellular proteins, oxidized cholesterols and cholesterol in atherosclerosis. And finally, macular degeneration is somewhat underappreciated storage disease where you get profound lipofuscin formation and these carotenoid lipofuscin components. And we tried pretty much everything, but this was the one that worked best, so I'm going to talk mostly about this and a little bit about other projects. So in macular degeneration, we tried to discover enzymes degrading this special carotenoid lipofuscin that you see there. The macula is a spot in the back of the eye that's where the light gets focused that comes through the lens. It's responsible for high acuity color vision. And as a consequence, when the macula degenerates, you lose the central focus of your vision first, and then the spot gradually grows to the outside, increasingly consuming your vision. And the process that's happening at the macula is that you have the photoreceptors that sense the light coming in from above, or hitting these things here, really, these rod outer segments. And they are intercalated with the retinal pigment epithelium, which do most of the metabolism for the photoreceptor cells. Most importantly, the vitamin A metabolism, where vitamin A, after absorbing a photon, gets recycled in the visual cycle in the retinal pigment epithelium. And as a consequence, the retinal pigment epithelium has all the lysosomes that do the processing of the junk molecules. When you look at the macula under visible light, you see that it's fluorescent. And that led to the identification of the fluorescent compounds. Unlike lipofuscin in other cells, the lipofuscin in the macula mostly consists of these vitamin A-derived compounds, and the most abundant compound is called A2E, and the best characterized one. A2E because it's from 2 molecules of vitamin A that form the chains, and 1 ethanolamine from the membrane. And this is the A2E formation pathway. It starts with all trans-retinal, an intermediate from the visual cycle up here, condensing with phosphatidylethanolamin in the membrane. First, 1 molecule of all trans-retinal will condense, and then exists a recycling system called ABCR4 that flips this to the outside where it gets degraded. So great, why do we have a problem? Well, because no system is perfect, and some all trans-retinal will always escape and will condense with a second molecule of all trans-retinal, and that leads to the irreversible pathway of A2E formation in the retinal pigment epithelial cell. There are heritable mutations in this transporter ABCR4 that cause, you can imagine if this transporter is mutated, you get no removal of the intermediate, you get much faster A2E formation, and that causes heritable early onset macular degeneration called Stargardt disease. People have no known enzymes to remove A2E, no known ways really to remove A2E other than photodegradation, which has its own toxic problems we'll learn about later. So it's an example of catabolic insufficiency in aging. A2E is toxic in various ways, inhibits lysosomal ATPase, can cause downstream lysosomal failure. It's like a detergent with these hydrophobic chains and a charge in the middle, so it can compromise lysosomal membrane integrity. And then it's phototoxic, it absorbs in the visual range, and these double bonds do the absorbance and cause epoxide formation, free radical formation. Very bad. Current therapies sound like jokes, but are not. If you're losing your central vision, there are training programs to train people to really use their peripheral vision better. And they have some success, but it's not the kind of solution to the problem that we're looking for. Also to mediate phototoxicity, glasses. No joke, that's truly being done. And then this is a better one. VEGF inhibition is only for the wet subtype. Remember, in macular degeneration, you have dry and wet macular degeneration. And in the wet subtype, for unknown reasons, you get blood vessel ingrowth into the macula, so the vision gets blurred further by the blood vessels. So, for example, Genentech's Avastin, the VEGF antibody, can help reduce this abnormal vessel growth. But that brings only the vessels back, that cuts the blood vessels back, but it still doesn't do anything about the loss of central vision that's due to retinal pigment epithelial degradation. So, to cut a long story short, we propose A2E as a novel drug target, a small molecule that we are targeting with enzyme drugs. Since we were doing stuff on the cheap, this was a great project for us because A2E is red, so when it gets degraded, it becomes less red. This is just a dilution series that we prepared. And this is how we identified our first A2E degrading enzymes. These two kids basically came up with the idea that we should be testing vitamin A degrading enzymes for whether some of them would also by chance accept A2E as a substrate. And as it turned out, most of them didn't. We screened like 20 of these. But this one, NSC1, you can see the color is markedly more pale. And that's how we first identified it. And then our collaborators at the universities used their high-tech gear to confirm the degradation. These are HPLC traces now done at a collaborating university at St. Paul, Minnesota. A2E runs as 2 peaks. It has an isoform, the iso A2E, that's kinked. So you always see 2 peaks when you look at A2E. And the average enzyme did absolutely nothing to these 2 peaks. But ours formed a new 3rd peak that's a product, completely degraded both isoforms of A2E. And you can see in the spectrum that this major toxic absorbance peak is lost. And with mass spec, we found the reaction that our enzyme catalyzes. It chops the long arm of the A2E molecule right in 2. Beta-ionone is actually a scent in rose oil, so I don't know if we have enough A2E in these patients to make them smell like rose oil, but that would be a beautiful therapy.